was in the okay, sorry. So welcome everybody, as I said, to the second seminar, which is part of the Aristo project. And so, as you know, we will meet 2 months, and this is now the second one, and we will have the chance to welcome Anne Steenberg. She's a policy officer at the Dutch board for the EU authorization of plant protection products and biocides. And the task of this board, as you know, is to assess whether plant protection product and biocidal products are safe for humans, for animals, and the environment, in accordance with a number of international regulation agreements and criteria. And so today she will give us a talk about biopesticides and risk assessments. And so we'll have the pleasure now to listen to Anne. So Anne, the floor is yours.
Well, thank you, and very nice to be here.
See if I can... Everybody's cut his micro in case of.
Yeah. Just let me see the window and then share, and then I hope you can see the slides again.
Great.
Yes, good. Well, I'm really happy to be here, and I'm hoping for a nice discussion also on topics related to risk assessment and microorganisms. I'm indeed a policy advisor at CTGB. So CTGB deals with plant protection products and biocides. And let's dive into the contents, I would say. So in this presentation, I will first discuss what are biopesticides in contrast to just normal pesticides. And we will look at differences in the risk assessment between conventional chemical pesticides and these biopesticides. And in a lot of cases, we need an alternative approach to perform the risk assessment for a biopesticide compared to these conventional ones. And when you have an alternative approach, then it may be difficult to harmonize. So how do you harmonize an alternative approach between all the member states? And then we will look at current and ongoing activities to improve the regulatory framework for biopesticides. So this is what I have prepared slides on. And as I said, I found it really difficult to prepare because I don't know about your background and how much you know about the regulation. So please feel free to interrupt if you have questions, or if you think, oh, we all know this already, please move along. Then please let me know. And also I have included some discussion points at the end. So it's not a full hour presentation, but there are some discussion points at the end. Well, to dive into the really regulatory bit of the risk assessment, in the EU, plant protection products are regulated, of course, and we have this main regulation, which is called the Plant Protection Product Regulation, which is often referred to as the 1107. And regarding the risk assessment, this main regulation just says when you use plant protection products, they should be sufficiently effective. So you should not use something that is not effective. And it should be safe. This is in Article 4 of this regulation. And related to the risk assessment, what is also in this main regulation are the approval criteria for the active substance. So the hazard properties that an active substance needs to meet to be able to approve this substance. But this main regulation doesn't say a lot about how to perform a risk assessment. So which information is needed, how do you need to evaluate this, and how do you need to decide whether this is indeed effective and safe. So for that purpose, there are implementing regulations. So they are implementing this main regulation. And these implementing regulations are called the data requirements. And we have data requirements both for the active substance and for the product. We will get to that a bit later. And there's a regulation that we normally refer to as the uniform principles. And these are the uniform principles for the evaluation and decision making. So in this regulation, you get information on how you should evaluate all the information that you have. And based on the information which you have and the assessment, what your decisions should be. So can we approve or not approve, or authorize or not authorize? Yes, so as you may have noticed, there's a distinction between approval and authorization and active substances versus products. And this is because in Europe, in contrast to many other regions globally, we have a two-step process. So first of all, to be able to place a plant protection product on the market, the active substance has to be approved, which is EU-wide. And once that has happened, so it's an approved active substance, then at member state level, products can be authorized. And most of the contents of this talk will deal with this first step, so the approval of the active substance and the assessment of the active substance. And this process is depicted in the picture below. So there's an applicant or a notifier, we normally call them, who submits a dossier. And they submit this dossier to one of the member states to act as a rapporteur member state. So there's one member state that takes on the majority of the work for this dossier. And this rapporteur member state prepares a draft assessment report. So based on all the information is evaluated, and there's also a proposal for a conclusion. Then this set is shared with all the member states and with EFSA. And this is the peer review of this assessment. And then all the other member states can comment. And then, of course, these comments go back to the rapporteur, and the rapporteur has to amend this draft assessment report accordingly. EFSA also gives comments, and then what EFSA also does is publish the conclusion on the peer review of this substance. So I don't know whether you are used to these EFSA conclusions. You can find them online. So for each substance, there's this EFSA conclusion. And this is really the outcome of the risk assessment. So this is the final stage of the risk assessment. And what happens afterwards is that all this information together is sent to the European Commission. And the European Commission are the risk managers. So next to the risk assessment stage, you have the risk management stage, and they prepare a review report. And this, together with all the information, goes to the standing committee where all the member states can vote whether this substance is approved or not approved. If it's approved, then this is also published, and then you can have these product authorizations at member state level. So this is a, a long process with all member states involved and EFSA involved. Yeah, the role of CTGB is, of course, we can be one of the rapporteur member states. And currently, CTGB is the rapporteur for 20 out of the 28 biopesticides that are currently under assessment. So we have, well, a majority of the biopesticides are dealt with by CTGB at the moment. Then to go to the next bit, to the definition bit. Well, talking about biopesticides in Europe is always a bit tricky because different people might mean different things, and especially globally, biopesticides is not interpreted the same way. And in the EU, we do not have a definition of biopesticides. So in our regulation, not in the main regulation and also not in the implementing regulations, there's no definition of what is or is not a biopesticide. However, we have a general understanding of what we consider to be biopesticides. So for example, no macroorganisms, so no parasitic wasps, for example, or these ladybugs, they are not considered to be biopesticides. And also no nature identical substances. So for example, if you have a plant metabolite, if you produce it synthetically in the EU, it would not be considered to be a biopesticide, whereas in the US it would be. Except for semiochemicals, which are, for example, pheromones. These are mostly produced synthetically, and they are included in the biopesticides. So generally, talking about biopesticides, we mean botanicals, semiochemicals, and microorganisms. And then, of course, things are always developing, and there might be new categories of substances. of which we can have a discussion whether we should consider these as being biopesticides or not. So RNAi, for example, antibodies or peptides, well, take your pick. They can be biopesticides or not. We do have some other definitions that are relevant in this context in our regulations. For example, we have 4 different types of approved active substances, which are the basic substances, low risk active substances, the candidates for substitution, and then there are the ones that have no, not one of these labels, and they are, well, the normal active substances. So whether they receive this label is the outcome of the risk assessment, except for the basic substances. Basic substances are a bit odd in the sense that they are substances that are used for something else than plant protection, but they might have their use in plant protection. So an example is, for example, when you use beer to attract slugs and catch them, then this beer is a basic substance which you use for plant protection, whereas it's marketed as something else. So a basic substance cannot be sold as being a plant protection product. It is something that you buy as something else, but you can use as a plant protection product. But you can only do so if it has been approved as a basic substance. So for a professional grower, for example, they cannot just use something in plant protection, then it would first have to be approved as being a basic substance. The other ones are really the outcome of the assessment. So whether something is a low risk active substance depends on the outcome of the assessment. So it can be chemicals, it can be biopesticides, they can both be low risk, and that's determined also as part of the assessment. And the candidates for substitution are kind of the opposite of the low risk active substances. They have certain hazard properties, and they can be substituted if there are sufficient alternatives. So then they're not allowed to be authorized anymore if there are sufficient alternatives. Then there are 2 other categories in the regulations. So in the implementing regulations, there's a distinction between chemical active substances, which are all assessed according to the part A's of these implementing regulations. And we have microbial substances, which are assessed according to part B. But because we only have chemicals and microbials, that means that all substances which are not microorganisms are assessed according to part A. So if you have a botanical, then you assess it according to the same data requirements and the same principles as you would for a conventional synthetic substance, and also for pheromones. So we only have these 2 classes at the moment. So concluding for biopesticides, they have no separate status in the regulations. Low risk does have a separate status, and it's really the outcome of the risk assessment. The botanicals and the semiochemicals, they follow the chemical assessment, the chemical data requirements. We do have separate data requirements and evaluation and decision-making criteria for microorganisms. And we have separate guidance documents. So for example, for botanicals and semiochemicals, we have guidance documents. There are also guidance documents for sections of the assessment of microorganisms, and we are currently working on an overall guidance for the assessment of microorganisms. Are there any questions up till here?
Maybe just a very brief and for the active substance, the nice schema that you show very at the beginning. So all the member states should agree, or can one member state not agree and anyway the product continues live? How does it work?
Well, voting is a majority.
Majority.
Yeah. However, yeah, normally EFSA has a big role in this peer review, and so you can sometimes there are issues that we justify that we don't agree with the proposed way forward. But still, yeah, it's a tricky business because you want to have as much harmonized as you possibly can. But most often this means having more data than you possibly would want to have. So going more in depth. So it's a balancing act, but it's not that everyone has to fully agree. And also there is sometimes a disagreement between EFSA, for example, and the rapporteur. And now there's the possibility to have this included also in the EFSA conclusion. So then there's EFSA's opinion, and then next to it, you can have the opinion of the RMS. And then it's for the risk managers to make sense of this and propose the way forward in the review report. And then there's the voting where you have the majority vote.
Okay, thank you.
Yeah. But it's indeed one of the most tricky issues of this alternative approach to harmonize. Yeah. This second bit will be more really about the assessment itself, more the principles of the assessment. So first of all, looking at conventional chemical substances, well, mostly the pesticides we used to know were synthetic toxic substances. And we know that these synthetic toxic substances have certain properties, certain hazards, for example, persistence and bioaccumulation and toxicity. They are very relevant hazards and very well-known hazards. So this regulation was set up to deal with these types of hazards for these conventional pesticides. But then for biopesticides, well, are they also fit for purpose for these types of pesticides, or do they have different properties? Well, sometimes people think, well, if it's biological, if it's green, then it should be safe because it's natural and we should not worry because it's just safe. But of course, that is not the case because, well, microorganisms, plants, they can all produce toxins. Some microorganisms can be pathogenic, for example. So a risk assessment is needed to determine whether these substances are safe. However, the hazards that apply to these biopesticides, so the inherent properties that can cause harm to humans, to animals, to the environment, may be different between biopesticides and the synthetic ones. And the approach by which you can determine whether they are safe might also be different. So in the next slides, I have two examples. One is about botanicals, so plant extracts, and the other is about microorganisms. Looking at the risk assessment for a conventional chemical active substance. Well, we get a dossier. On the dossier, it says, this is the dossier for substance A. And substance A, when you look at it under with a HPLC, it looks like one peak, one component. And we know this substance will be used, for example, 1 liter per hectare. And then we can calculate, okay, this substance and this amount, then the exposure of surface water will be, well, we have a model to determine, or leaching to soil or dietary exposure. So we know what the assessment should focus on, and we have the exposure models to determine the exposure of humans, animals, environment for this one component substance. When you now look at biopesticides, then mostly they have this inherent complexity. Well, immediately you see several peaks. So which one of these peaks is now relevant? If I want to do residue trials, for example, for dietary exposure of consumers, what should I measure? Should I measure all peaks, or perhaps is a single peak? Is it most important? So this dossier doesn't say substance A and we can focus on substance A, but it says plant extract A. But what should we focus on? So there is an additional step beforehand. And of course, just knowing that there are several components in this substance doesn't say a lot about the risks. They can be very harmless components. So you don't know by looking at this chromatogram whether this is lattice and we should not worry about all these peaks, or whether there's a very potent toxin in amongst these peaks. So you need first to determine which of these components of this substance is relevant for the risk assessment. Also, botanicals can be very diverse. So you can have substances such as spearmint oil or garlic extract, which are complex, but well, perhaps you need not worry too much about individual components. And you have also the ones that have really a toxic mode of action, such as azadirachtin or pyrethrins, which are more complex. And then, well, how to deal with this in the risk assessment. Luckily, there is room to do things differently for plant extracts. So they are regulated according to the chemicals, but in the data requirements, for some of the data requirements, there's this phrase saying, for plant extracts, a different approach may be taken if adequately justified. So you're allowed to do stuff differently as long as you can justify why this is appropriate for this substance. So reasons for having a different approach might be that you know that this substance occurs naturally because it's a plant commonly grown in the EU, for example. There might be other uses, especially food. For all substances, there's the possibility to waive studies, so to not include studies in the dossier if this information is not needed for the risk assessment, or if it's not possible to generate. So then you can also justify why you have not submitted a study. But this all means, again, that we have this customized risk assessment. So when the RMS determines, oh, okay, that's only this one component that is relevant, and I'm not going to do anything for the remainder of the components. Well, how do you harmonize this within the EU? If another country says no, but I think we should include this other one also, then of course you have a problem because it's very late in the process, and then additional information needs to be generated. And so how do you agree beforehand on having a common approach? It's really difficult. For example, we were the rapporteur for rapeseed oil, which is, well, it's triglyceride. So there are common primary metabolites, just normal building blocks of plants. We all know, I think, these yellow fields in Europe where it's only rapeseed growing. And the active substance was food grade rapeseed oil. So you can just use it as human to consume. It was safe food grade. And then looking, for example, at the environmental assessment, so the exposure assessment, yeah, you might wonder, do we really need to have information on the rate of degradation of this rapeseed in soils? And do we need to know about the root? So for synthetic chemicals, it's really relevant to know whether there might be some degradation products that have, well, perhaps different properties than the parent substance, or perhaps are very persistent. So you want to know, okay, if we put this substance in the environment and it's a foreign substance, it's not a natural substance, we want to know whether it degrades. But of course, for triglycerides, the situation is a bit different. You might also wonder, okay, do I really need all these models that we normally use to determine whether this rapeseed will leach to groundwater when we know that it's cropped extensively and people are eating it? And do we really need to worry about rapeseed popping up in our groundwater? The applicant had submitted information on degradation in soil and other information that we considered, well, not relevant for the risk assessment, because without this information, we could also perform a risk assessment for this substance. Still, what you see in the EFSA conclusion is always also a list of data gaps. So data gaps are areas where the data requirements have not been met. So a study is asked for, it's not in the dossier, then you get a data gap. And in this case, EFSA labeled them as not critical, but relevant for the assessment. And these are just a few examples. The list is longer. But for example, you get data gaps for a validated analytical method to determine the rate of chemical degradation of rapeseed oil in aerobic soils. Also, a detailed summary and evaluation of a degradation study, again, in soil. The calculations of the predicted environmental concentrations using a certain model. And also, it's not to do with degradation in soil, but still it illustrates the point, I guess. An assessment of the potential effects of rapeseed oil on the biological methods for sewage treatments. So if I use rapeseed oil in my crop, will this affect the microbiology of the sewage treatment, so the municipal sewage treatments. Well, I don't know whether this is really relevant for the risk assessment. So sometimes you choose an alternative approach, but then there are different opinions. And in the end, the substance is approved, but it takes more time. And it might also lead to question marks at product level. So it just makes the whole process of approval and subsequently of product authorization less efficient if you have this discussion. So how could we improve this regulatory framework? And this is also where Ration, so the other Horizon project, comes in. Well, the main thing that we need to do is to find a way to harmonize these customized approaches. A way to do this could possibly be to make a proposal for the revision of the data requirements and the criteria for the evaluation and decision making. So if you can say beforehand, okay, this is a primary plant metabolite, then in this case, you don't need to provide information on the route of degradation, for example. So this could be in the data requirements. What also would be very helpful is to have a guidance, to be able to identify which of these components in a substance is relevant for the risk assessment. So, you know, there are all different components, and usually you don't have all the information on all the components. So you need to know about the source of the botanical, what is the plant, what can it do? And then you need to know, well, do I need to be looking for components? It's not that you have a list with all the data and that you can just decide, but we need to have guidance to identify which components are relevant. In some cases, it would also be helpful to have more test guidelines. For example, for poorly soluble substances, well, if something doesn't dissolve in water, how are you going to test for ecotoxicology, for example? And this will all be points that might be addressed within the Ration project. So those were the botanicals. And in the next few slides, I will spend some words on microorganisms. And I don't see any hands. I don't know whether you can raise hands in Zoom, but if there are no questions, I will just continue. Good. Well, of course, as you know, microorganisms are usually diverse. And for plant protection products, it's mostly fungi, bacteria, and viruses that we deal with. And of course, there are very fundamental differences between a conventional synthetic pesticide and a microorganism. Well, one of the most obvious ones is that microorganisms can multiply. And for the risk assessment, this means if I have this synthetic chemical, I know it's 1 liter per hectare, and I can determine what the maximum exposure will be of humans or the environment to this substance. For a microorganism, of course, they can multiply. In most cases, they won't, but they can. So you don't have a hard upper limit for the exposure. And as a risk is always the hazard times the exposure, you don't have a fixed upper limit to determine whether there's a risk. So this is a really big difference between synthetic chemicals and microorganisms. And of course, a second big difference is that these microorganisms, well, yeah, they produce all kinds of secondary metabolites. And of course, secondary metabolites are chemical compounds, but still you cannot use the framework we use for chemicals to the metabolites of microorganisms. So the next few slides I took from a course, a Better Training for Safer Food course on the risk assessment of microorganisms. And there we look at what are actually the hazards that apply to microorganisms when you use them in plant protection. And well, most people, when they think about microorganisms, they think about disease. So pathogenicity, infectivity is of course a hazard property of microorganisms. And the second one is toxicity, not of the microorganism itself, but of the metabolites it may produce. And for today, we are only going to look at those two hazards. So thinking about the first one, to be able to assess whether a microorganism is pathogenic, well, yeah, of course, there is a difference between this assessment and the assessment of a chemical, whether this is toxic. But there is also a more fundamental difference, which is that for chemicals, we have test guidelines. So we know how to use test animals, so animal models, to test for toxicity. And then we know how to translate these effects that we see in test animals to the effects that we expect to see in humans. So we have the test guidelines and they are appropriate for active substances, and we can translate it to the possible effect to humans. When you now think about a microorganism and you want to assess whether a microorganism is pathogenic, then an animal model may very well not be appropriate. So, and I think this is a really nice picture of a researcher, and she's looking at sputum, so spit samples of patients, and she needs to determine whether these samples are from patients with tuberculosis. And the way she's doing this is, I don't know whether you can see on the screen, but this is a rat. And she has a sniffer rat that will sniff all the samples and then will indicate, this one tuberculosis, and no, this one no tuberculosis. And the reason why this works, well, first of all, this rat has a very acute sense of smell. So it's like a sniffer dog. It can smell, sense very well. But secondly, it works because this rat is much less susceptible to tuberculosis than humans are. Otherwise, she would have to train a lot of new rats every time because they get tuberculosis. So if you would use a rat to test whether tuberculosis is pathogenic, you might come to the wrong conclusion. And in the case of tuberculosis, it's just the case that the rat is less susceptible. But very often, well, it's just about the host range. So if the rat is not in the host range and we are, or the other way around, well, then you get to the wrong conclusion about pathogenicity. So how do we do this in the risk assessment? If we cannot use an animal model to determine whether it's pathogenic, this does mean that we cannot determine whether this microorganism is pathogenic. Well, no, luckily this is not the case. We are capable of determining whether a microorganism is pathogenic, but what we use is not guideline tests to determine, to screen for pathogenicity, but looking at the information that we have on this species of microorganism, on its close relatives, everything there's known about these species, and then you can draw a conclusion. And in the working group where we worked on the data requirements, there was a Swedish professor, and he said, well, if you find an unknown human pathogen, you'll get the Nobel Prize. And of course, he didn't mean that if you find some strain of a microorganism that, well, this strain we didn't know, but the species we did, you'll get the Nobel Prize. You'll have to do better. You'll have to find something that, based on the knowledge that we have about this species or its close relatives, we would not expect it to be pathogenic, but it is pathogenic. Then this is so unexpected, you might very well get the Nobel Prize. So based on the information that we have on the relatives, you can determine whether you should be worried about pathogenicity. And then, of course, in some cases, the outcome is, okay, there's no indication that this microorganism will be pathogenic. And in some cases, there will be indications. So for example, for Bacillus species, they are, of course, closely related to pathogenic organisms. So in case you have indications that there are close relatives that are pathogenic, then of course you would want to have information at strain level to see whether these virulence factors that make this microorganism pathogenic are present in this strain. So if you have indications, you might need information at strain level, but no standard guidelines that you can use to screen for pathogenicity. Then the second hazard is toxicity. So toxicity of the secondary metabolites that are produced by microorganisms. And again, for chemicals, it's also toxicity, it's also chemical substances. We have the test guidelines, they are appropriate, but the big difference here is that we know if we get a dossier for substance A, we need to test for toxicity for substance A. But when we get a dossier for microorganism A, Okay, so which of these metabolites are relevant for the risk assessment? Are there any or are there none? How do you determine? And this is especially difficult because, well, they might produce a lot of metabolites. So, for example, for streptomyces, there are, and I think it might be higher today than it was at the time that I made the slides, 19,000 known metabolites. How do you determine whether there's one in amongst them that you need to worry about? So it's also just more complex than for chemical substances. On the other hand, microorganisms and toxicity, well, we have this huge challenge, huge numbers of metabolites. Some we know of, some we don't know of. And also there's no extract of metabolites of species A that we can test for toxicity because they don't make these metabolites all at the same time because they only make them when they need them. So you don't have a test item that you can just use your normal toxicity tests. So this is a huge challenge. On the other hand, when we think about, and this is where you know a lot about, of course, about our microbial world and how many microbes there are everywhere. I mean, 1 billion in each gram of soil, they are all producing metabolites. And when we're thinking about food safety in the EU, we only have maximum levels for 6 groups of microbial toxins. And of course, for human pathogens, because they also might produce toxins. So there's this seemingly contradiction between the huge scale of the number of metabolites that a microorganism can produce and the numbers that are interested, they are interested in when it comes to food safety. But still, they have to be included in the risk assessment because some of them are really potent toxins. And also, I talked to a couple of former colleagues of mine who are all soil microbial ecologists, and they said, oh, you should not worry at all about the production of metabolites. And every gram of soil, 1 billion of microorganisms, who cares about the metabolites that they are producing? And then I said, yeah, I know there are a lot of microorganisms in soil, but well, what also happens, this is an example from Bayer, is that they spray on crops, for example, strawberries, just before they are picked and put in a plastic box and sent to the supermarket. Or on a cucumber because before the nice plastic wrapper is put on and then sent to the supermarket. And then they said, well, no, but you should not do that. You should not spray microorganisms on the stuff you are going to eat in a bit because you don't know. I said, well, yeah, but that's what we do. So we have to assess whether this is also safe. Can you spray this on strawberries and then sell it? And then there will be all kinds of people, also children, eating the full box of strawberries at a single time. And is this safe? And of course, there are also a lot of microorganisms on the strawberry that you normally eat. But how do you know whether this is also safe? So it's a tricky question. So for microorganisms, for their metabolites, they may be toxic. They might be relevant for the risk assessment, but we cannot ask data for 19,000 metabolites for a single microorganism. And of course, we also don't need to do this to conclude whether this is safe. However, it also means that we have this additional step in the risk assessment. As for the botanicals, where you need to determine which component is relevant, for microorganisms, we also first need to assess what needs to be assessed. So are there any metabolites that can be produced by this microorganism that really present a foreseeable risk? So something that we really need to worry about. And this is where something called problem formulation comes into the risk assessment. And if you're interested, this is a really nice article on problem formulation in the context of GMO, but also related to microorganisms. So you have this extra step compared to chemical substances. And well, this demonstrates that there is really a difference between chemicals and microbials. For chemicals, all the data requirements that we have are relevant. And if you just follow them, the data requirements themselves will say when you need more information on or not. They might say, in case there's more than 10% of this, you need to supply this data. But of course, For microbial active substances, we do not have such a 10% because they might grow, they might produce stuff. So we cannot use a quantitative approach. And even more, to make it more difficult, there is this huge diversity of microorganisms. It can be an entomopathogenic fungus, it can be some bacteria working through competition, it might be a bacteriophage. So how do you determine what is needed for this specific microorganism? So you need to have this explicit problem formulation step in your risk assessment. And what you use is all the information that you have. So on the identity of the microorganism and its close relatives. You use preliminary data because the applicant has selected this microorganism, of course, because of its properties. So it's relevant to know what these properties are. And also in the data requirements for microorganisms, they have just been revised. And in these new data requirements, in 3 places, it's not called a problem formulation step, but in 3 places, the data requirements ask for the outcome of a problem formulation step. So for example, for the metabolite assessment, there's a data requirement that says, please indicate whether there are any metabolites that are relevant for the risk assessment. And then, of course, you need to provide information why there are no relevant, in most cases, metabolites. So based on which information can you determine that you don't need to submit any further data on these metabolites? And the same is the case for the chapter on human health, where you also need to submit a statement saying, okay, based on the body of knowledge, it's not pathogenic, for example, and we have not submitted further information. And the same is for the ecotox section. So first gather all information and then determine which tests or information are needed for this specific microorganism. And of course, there is, well, the big hurdle again. You can do this for yourself, determine, okay, well, I have this information, so I'm going to take this route. But then how to harmonize this problem formulation step between 27 member states? Is really difficult. So we need to have additional guidance on how to perform this problem formulation step. And also for microorganisms, and especially for the ecotox section, we need test guidelines. We don't have any test guidelines for microorganisms. We don't know how to test definites for the effects of using a microorganism. There are also other ways to improve the risk assessment, for example, regarding the procedures. Currently, when we get a new strain of a certain microorganism, for example, the Bacillus amyloliquefaciens that we just seen, if we get a new application for a new strain, we start all the way over again from scratch, trying to get this body of knowledge, which is a lot of work. So we could look at ways how you can use this information at species level also for new strains. And also, it's good to remember that for all plant protection products at the moment, we really need to have a more efficient assessment because capacity of assessors, both at member states and at EFSA, is really a bottleneck for the assessments of active substances. And well, it's now limiting the innovation of pesticides, but it's also limiting the reassessment of old pesticides. So it would really increase the level of safety if you can reassess old pesticides quicker and approve new pesticides sooner. So more efficiently perform this assessment. So what is being done at the moment to improve this regulation of biopesticides? Well, currently there are several projects ongoing. Some are at OECD level. So OECD has an expert group on biopesticides, and they are going to start working on test guidelines and are also preparing consensus documents for microbial species. So for example, for a Bacillus species, have a consensus document, and then you can use this if you have a new strain. And they have recently published a guidance specifically for the assessment of bacteriophages, so viruses that infect bacteria. And for these test guidelines, there is a link between the OECD work and the RATION work, because this is also where ration can contribute. Then there's the EU Working Group on Biopesticides. They have recently worked on the revision of the regulations, the parts of the regulations specifically for microorganisms, and are currently working on a document which is called the explanatory notes, and which also might be interesting for you, because this will describe, well, provide explanation for these regulations on microorganisms. So per data requirement, there's information on why we need the data, how we assess the data, and what the outcome can be. Currently, it's under in the commenting phase within the working group, but we have prepared this document. So CTGB has prepared the document together with the Danish EPA, and our version, which we have delivered to the Commission, is already present on our website. So if you follow the link, you will get to the CTGB Danish version. And hopefully, well, perhaps after the summer holidays, the EU version can be endorsed at scope-of level. Yeah, then this EU working group has also worked on guidances for metabolites and guidances for antimicrobial resistance. And of course, there is the Ration project, which is really aimed at improving this assessment for biopesticides, and improving also includes making more efficient. So it's really aimed at improvements which are easy to implement. Well, I don't know whether you know about the policy goals in the EU, but well, they have high standards for reducing the use of chemical plant protection products. But then, of course, we need to have alternatives. And the sooner that these alternatives enter the market, the better. And currently, it's just taking a long time. So this is the end of the bit I prepared, and it took a bit longer than I expected. But anyhow, if you have questions or remarks, I'm very happy to hear them. I also need to put CTGB in the spotlight, because we are always looking for people to join us. And in case we have time left, I have some discussion topics on the next slide. But first, I'm really curious to see whether you have any questions or remarks.
Thank you, Anne. So the floor is open for questions. Maybe I can start with a brief one. We know that the registration for biopesticide is a very long process in Europe. It can take several years, much more than in the US. And we know also that there is this other regulation for the biostimulants, and some microbes, as you know, can have dual properties as bioprotection and biostimulant. So most people go to the biostimulant because it's much more easy. But the regulation is less complex, less tests, etc. How do you deal with this? Is there a consensus between different commissions? How does it work?
Well, the way it works is that biostimulants are to be regulated by the Fertilizer Act.
Yes.
And plant protection products are regulated by the Plant Protection Product Regulation. And well, I was told when. Yeah, no, I know. I think I know what you mean. When I joined CTGB, one of my colleagues said, "Well, yeah, but you have to know, there's regulation and then there's reality. And in the world of regulation, what you write on your on your product. So if you say my product stimulates resistance against drought, then it's an abiotic stress and you're a biostimulant. Yeah. And if you put on your product, this works against aphids, then you're a plant protection product. So really the claim on your product determines whether you go left or right. And of course, when there are obvious signs that something says it's a biostimulant, but actually it's being sold and promoted as being a plant protection product, well, yeah, then there's somebody that said, no, this should not be. So we know it is happening. Yeah, it's really the claim on the product that determines.
Yeah, but you mentioned the case of streptomyces, which is a biopesticide. but which also is a microbe that also can play key roles in plant nutrition. So myself, could I choose to go with a streptomycin species to the biostimulant, or
Yes.
would people tell me, oh no, stop, this is really bioresistant, whatever you tell?
No, it's really the claim that you put on your product. So if you say this one is good against drought, or it helps phosphorus acquisition, biostimulant. But if you say it helps against plant pathogen X, then it's a plant protection product.
Okay.
So it's really the claim that you assign to your product. It's not that we have lists of species or anything.
Okay.
Yeah, I saw Dimitrios has it.
Yes, Dimitrios.
Yes. Just to spark a bit of discussion. So thank you for the very nice presentation. I have some questions about the presentation, but also some questions about your personal career and development, because we have students in the audience that they are now at the end of the second year of their PhD, and in 1 year time they will be looking for opportunities in the market. So I think it will be important to talk about this as well. So my first question is about pathogenicity and infectivity, because information about the potential of a strain to be a potent pathogen we could also get from the whole genome sequence as well. So I'm not sure how much is expected, because we've been discussing a lot about this in ration, and we really think that whole genome sequence analysis can serve a lot of purposes in pathogenicity potential, in infectivity potential, in AMR, in antibiotic microbial resistance, in secondary metabolites. The tools are there, but I think we need to end up with to help the regulators and the people that they will assess. We need some really simple tools that they will give an idea to the regulator if this is potent or not. Something like the focus models. Okay.
They are really simple.
Well,
no, I know what you mean. Yes, and well, two parts of the answer. So the first one is that for some microorganisms and for some questions that you have about microorganisms, genome sequencing is the way to go. So for example, for antimicrobial resistance, where your hazard is a gene which is present or not in your microorganism, of course, genome sequencing is the best way. And this is also the way that is described in the guidance. Regarding the production of metabolites, if you are worried that your strain will produce a toxin because you know that close relatives produce this toxin and you know the genetic basis for this toxin, then it's really relevant to check the genome to see whether this is there. And the same for virulence factors. What I think is the biggest hurdle to use whole genome sequencing now is that the annotation is not always up to scratch. And so it's done by analogy, and then it might say it's something really scary while it's not. So we don't know just from the sequence what the gene is doing. And even if we would know what the gene would be doing, then is it expressed? We don't know. So in a lot of the cases, we have the feeling that it would make the assessment more difficult, but not yet inform the assessment better. So you will get all these red flags saying there might be something the matter here, but we have no ways to determine whether this is actually the case. Part B of the answer is that EFSA is currently working on a pipeline for genome sequencing. And they are also, first of all, focusing on AMR, for example, but then are going to also add known virulence factors and known metabolites. So yes, genome sequencing is, I think, the way to go, part of the way to go. But I'm really hesitant to ask genome sequencing as a screening method, because it might show you all these things that might happen, but are really not relevant for the risk assessment. So in specific cases, it might be really helpful.
Yeah, it can give you some answers, but not all the answers. I mean,
Yes.
But certainly it needs a lot of curation, I would say, at this level and stuff. This is a common problem that we are facing with bioinformatics at curated databases.
Yeah. And then that's not always obvious to everyone that this is a bottleneck. And then they think, okay, but then we can just use this. And then the assessment gets bigger and bigger and bigger. And it's not really, really helpful. But in the future, yeah, sure. I think we might go to rely much more on genome sequencing.
Okay.
Yeah.
Any
Are there any other questions? Otherwise,
I have other questions, but I will leave the audience to answer. To ask questions before.
I have a question.
Yes, please go
related
ahead.
with microbes. So in your presentation, you talk about some aspects that should be considered, such as pathogenicity, the production of secondary metabolites. And how about the persistence of the microbe in the environment? Because you're introducing like a foreigner like species, and how you track the persistence. And yeah, so this is like the first question.
Mm-hmm. Well, that's a very interesting question. And in the regulation that we used to have until, well, this year actually, there was a data requirement for persistence. So you had to demonstrate that your microorganism would return to background levels. And if it would not return to background levels, you would have to demonstrate that this is safe, that it has a higher level upon application. However, well, thinking about microorganisms, to start with, you have a strain of a microorganism, which is something you have given this given name, it's a strain, and then you go out into the field and you apply this. So by definition, this strain is something artificial, but of course it might be very closely related to organisms that are already present in the soil. And also, well, microbial communities, they are so diverse and the competition is so fierce in soil or on leaves or in the rhizosphere that we hardly ever see that the microorganism is still detectable after a year. It's sometimes we think it's a miracle that it actually does something because the competition is so fierce. So in the new data requirements, the data requirements purely regarding persistence have been removed. We don't ask anymore standard for data to show whether it goes back to background levels, whatever they might be. However, specifically in the case of microorganisms which are pathogenic, so pathogenic to, for example, insects, or pathogenic to aphids or something, or perhaps in the future to plants. We don't have any herbicidal microorganisms approved yet, but in those cases, when the microorganism has not been isolated from the EU, then we ask for additional data. But then this additional data really focuses on the hazard properties. So what is the host range of this microorganism? And does this strain cause effects that are different to, for example, the effects of closely related organisms that we know already occur? So it really focuses on the hazard properties and not so much on the population dynamics upon application. And then I'm curious to know what you think about this.
Interest. Okay, and I have a second question related also with microbes. So like you said, the soil, like if you think in soil, introduce this microbe, the soil is high diverse. So probably this microbe is going to like disappear like in, I don't know, like weeks, maybe months. Mm-hmm. But there is like some like papers that they talk about the impact of failure. So introduce the microbe, there's an unintended impact, so like a plant growth or like plant protection. But after a while, there is like some change in the microbial community or maybe change in the functioning. So how you take these assessments for like it's a month or like a year?
No, we don't. We don't take it. Well, I don't know whether I have this slide still. Yes, I still have. I don't know whether you still see it, but I have this slide on, well, this happening in an agricultural system. Yeah, well, this is a microbial hotspot being dropped on your agricultural soil. So yeah, of course, everything will have an impact on your microbial community. If I walk outside and I walk on the grass, then the community in the grassland will change because I've compacted the soil. And when it rains, it will be very different to the day before when it was drier. So everything will have an effect on the microbial community. The difficulty is, however, to know how this difference in microbial community, well, which is very redundant in its functioning, will affect the functioning of the soil. So for chemicals, we only look at carbon cycling and nitrogen cycling, which are, well, the most redundant systems microbial-wise in soils. And we don't look at community structures for chemicals. So I think to look at the change in the community structure because you add an additional microorganism, it doesn't tell you anything about the risks. You might even argue that this microorganism is a non-target organism because it's a naturally occurring organism that is present in the soil. So I think the situation for microorganisms and their effects on soil organisms is very different to the effects from chemicals on soil microorganisms, where I think more should be done. For example, to look at disease suppression or other ecosystem functioning. So just an effect on an ecosystem structure, on a community structure, I don't think tells you anything about the risks. And also in this case, I'm curious about whether there are any reactions to that.
For me, it's okay. I thank you for answering the questions. It was like really good.
You're very welcome. And I'm sorry we can't do this in person somewhere. It's always easier to have discussions.
Are there any other questions or remarks or points you want to address? I see an hand raising. Yes, Demetrius.
Yes, just to ask about, because I noticed that you had in your slide, but you didn't actually expand a bit on it about the QPS method, the approach.
Yes.
The qualified presumptions of safety, because it is used in other regulatory frameworks in food and feed additives, and there are thoughts, as far as I know, in EFSA of expanding this to other regulatory frameworks, but not yet to plant protection products. And I was wondering, what is your view on this approach being a potentially used in plant protection, microbial plant protection products, I would say?
I think it would be perfect. So what the QPS panel does is review I think twice a year, about 6,000 papers on microorganisms. And based on all this information per species, so it's not at strain level like we do for the regulation, but at species level, they say, well, this species, we think it's safe, or this species, we don't think it's safe. And we do make use of this information in our assessments. However, we cannot apply it to all aspects of the assessment. So it's only regarding dietary intake for humans. That's the only part in the assessment where we can really use the QPS status. And then it's the case that the outcome of the QPS assessment is very often this organism has the qualified presumption of safety. However, you should check for its toxigenic potential, for example. So check at strain level whether it produces any metabolites that are toxic. And that leaves us with having to do this whole metabolite assessment anyhow for a strain of this microorganism. So I have had a chance to talk to the head of the QPS panel and to discuss this and to, well, in my opinion, the way forward, if it's possible, would be to not only include in the QPS status human dietary intake, but also expand to the other areas of the risk assessment, but also the way that the information is available of the outcome of the assessment. So, for example, thinking about a Bovaria that is used as plant protection, then we know, okay, this is a Bovaria. Please check the levels of bovaricin in your active substance, because this is a metabolite that is considered relevant for the risk assessment. Instead of saying check toxigenic potential, because then we have to do the full metabolite assessment again. So I see huge advantages of having this first assessment performed by the QPS panel. But yeah, it will be a shift of the division of labor in the EU. And I think in the end, it will free up a lot of capacity. Also for EFSA, because they have now to check for each strain for plant protection products, instead of doing it at QPS once. So it's a very nice idea, but it's not in progress yet. And what are your thoughts? Would it be a good idea?
Yeah, I fully agree with you. I think it's a great idea because it will release a lot of, as you said, a lot of capacity, human resources for, you know, going through endless process of risk assessment that sometimes it's unnecessary.
Yes, yeah, I agree.
And there is the knowledge is there. I think the science to back it up is there.
Yes, yeah, I think so too. And also sometimes in the plant protection panel, people are still a bit chemical oriented, which is not strange because they have been doing this for a long time, of course. Whereas in the QPS people, panel, there's more microbiologists, and so it's a different background and also a different way of working. I see.
Your last comment actually, I think it's very relevant to ask about the capacities and the capacities of regulators at the member state level. If because you know we have here 9 fellows that they work with on microbial ecology, and I would like to hear your thoughts about because you are a microbial ecologist by training, okay, and you are working in a regulatory body. And now, as you mentioned, there is a decision made now in the European level that we are going to support biopesticides, and we would like to have them in the market as soon as possible and unblock that reach in the market. So I would think that we need in the regulatory framework people that they have good knowledge of how microorganisms behave in the natural environment if you release them and have a really good background in microbial ecology. So what is your thought about it? Do you think that at member state level, there will be a lot of opportunities for people with this expertise?
Yes. Well, there should be in every member state a lot of opportunity. And I think this is also happening. I've been giving these better training for safer food courses, and I've been noticing that every session there's more microbiologists in the participants. So I think there is already a shift towards people with more a biological background than a chemical background, and I think it's really a relevant job to do. And to be honest, when I joined CTGB, it was because, well, my daughter was just born and I was just so tired. And I thought, oh, science, I cannot do this at the moment. I'll do something easy. I'll go for a couple of years to CTGB and learn some stuff. But actually, it's really interesting. And so it's far more interesting than I thought. And it's really useful work because you really make this change in agriculture possible by having these alternatives ready on the market. So I hope there's more people with a background in microbiology in all member states. At CTGB, we already have a lot of colleagues with a background in microbiology, but are always looking for more. And I know that the Commission, the EU Commission, has also recently has a grant of 10 million euros for member states to increase their capacity. So also for biopesticides. So there should be also opportunity at other member states to join and to work on this risk assessment for microorganisms.
Excellent. Thank you, Anne.
Okay. Are there other points to address?
Yeah, see, there's a question in the chat still. Yeah, shall I read it?
Yes, please.
How do you deal with the complexity of identifying active compounds of plant extracts and registering plant extracts as a PPP? Yeah, as you showed in your presentation, plant extracts have different peaks, whereas conventional chemicals only have one. Additionally, these peaks of compounds in specific plant extracts may change depending on the environmental factors, and perhaps time conditions when harvesting the plants. Yes, microorganisms can also promote production. Yes, all true. Does this create regulatory barriers? Yes, it does. Yeah, so it does. So we have first a job to determine which of the peaks is relevant. And well, in some cases, it's obvious because we know about this plant and we know then about the mode of action. So we know what we are looking for. So in some cases, it's easy to determine, okay, these are the relevant components. In some cases, it's more tricky, and especially when we think it's not relevant, how to convince all the other member states and EFSA that it's not relevant. So the gray areas are always the most difficult. And then related to the differences in your plant extract between the years, between harvests, between I don't know what, for all active substances, you have to provide the specification. So this is the specification says, okay, this active substance should contain this and this amount of this substance and this amount of this substance. And usually for plant protection products based on plant extracts, it's a range. So for stuff that is causing a risk, there's, for example, a maximum. So component A can be in there for a maximum of this and this milligram per liter, for example. And for stuff that is useful for the efficacy, there might be a minimum. So then it says this component should be present at a minimum of this and this amount per liter of your extract. And this should be also incorporated in the quality control by the manufacturer. They should be able to demonstrate that each batch complies to this specification. But indeed, this is a barrier. So the easier the substance is, the easier it is to register, is the truth. I don't know whether that has answered your question. Yes, you're very welcome.
Okay. I thought that you have still a slide to show, if I remember well.
Yeah, I have a slide with things to discuss, but I don't know whether there's time. I have to pick up my daughter in a bit from school, so that's a deadline. We still have a couple of minutes.
5 minutes left.
Yes, 5 minutes I do have. Yeah, so if there is something of interest, then
okay,
please go ahead. So I have put down different subjects, for example, the production of antimicrobials by microbial pesticides. So is it a good idea to use a microorganism as a plant protection product when it's, for example, producing an antibiotic that is used as human medicine, so to treat infections in people? Well, the other question mark is, should we see the microorganism as the active substance or as a non-target organism? So this is again the discussion, is this something, should we see it really as a non-target organism that is present in the soil and we like it to be there? Or do we need to know indeed about persistence and whether it will go back to lower levels? We also already briefly discussed the community structure versus the function of a microbial community. And I think very interesting, well, to me personally, is the effect of chemical plant protection products on, for example, disease suppression. So for chemicals now, we look at nitrogen cycling and carbon cycling. Well, I guess in most cases, the microorganisms in the soil will go away when there's a pesticide that kills a lot, because then there's more carbon and nitrogen. But looking at disease suppression, so if you have soils that are disease suppressive, which is something that, well, have the policy goals, sustainable use, IPM we want to have, can we test chemicals, whether they affect this function of the soil to be disease suppressive? Is this something that could be incorporated in the chemical framework? So I don't know whether there's any discussion point to your liking.
Quite complex questions.
Yeah, it's for the Friday afternoon.
In 2 minutes.
Yeah.
I can sort of, give it a go for some of them at least. So I would start from community structure and function. There was a bit of a discussion about it, and I think at the moment with the tools we have in our hands, their level of standardization, the obvious answer is function. Well, community structure, as you mentioned, we don't know yet from by doing some simple analysis and saying, yeah, okay, the community changes. What does it mean? So we need to actually dive a bit more on this. And although the tools are there to get a very detailed analysis of what is happening in the community and in the individual members of the community, first, there is an obvious lack of standardization in the bioinformatic parts of it, because the rest of the analysis is pretty much standardized, I guess, because there are standardized protocols and a lot of things. But the pipeline of bioinformatics is, I think, it's a bit lagging behind. And of course, we need to put it in an ecotoxicological context to be able to provide some quantitative measurement or an endpoint. What does it mean? How much? What is the concentration level that affects 95% of the microbes there and leaves intact, and affects only 5% or 1%? So I think that's the main issue that we have to work with regarding community structure effects. Regarding the effect of chemicals on disease suppression, I'm not sure, because I don't know how many soils, if the soils that we can call them suppressive to a disease, how widespread they are, and if they are considered sort of a minor, not the exception rather than the rule, how much we can invest on it.
I'm sorry, I was going to say it depends on the disease also, I guess.
Yeah, of course.
So for some diseases, it might be quite common, but it's nice and easy to test, because you just need to have the soil and plants that are susceptible to the disease, and then with and without the pesticide. So it might be something else compared to carbon cycle and nitrogen cycle that you can just run tests for.
Yeah, you can dive in and identify tools that they could give you an idea of how you define these effects on the suppressiveness.
Mm-hmm.
Maybe an additional point, which is also of interest. People are more and more working on full microbiomes, not only on single microbes. So how do you consider this in registration? We can imagine that different microbes can work synergistically, can be used as a microbiome inoculant.
Mm-hmm.
It's something that starts to be considered, or not at all? It's too complex, or?
It is even more complex. We are starting to consider it because we have also made the opportunity in the new regulations to have a consortium of microorganisms approved as a single active substance. So this was not possible beforehand, and it is now. However, well, listening to people from research and development, I don't know whether we will be getting real consortia soon. So where microorganisms really have different functions, and because it's difficult to have a stable consortium. And so I don't know whether we see them for viruses, for example, where you have mixtures, but true consortia, I don't know yet.
But it's even more than consortia, it's really microbiomes. I mean, 95 or even 99% of the microbes cannot be grown. And
I know, yeah.
can play key roles in biocontrol, in biostimulation, etc. So I think that's also a point, which is probably the more complex of all, but that deserve also attention. There's been a paper recently in Nature dealing about the use of microbiomes, so complexes of microbes for disease management of biostimulation.
Yes.
So I guess this will also become a topic in the coming years or decades.
I don't know whether you want to have them regulated or go through another route and have soil transplantations, for example,
it's kind of
similar to the poop transplantations.
Yeah, exactly. That's what I was thinking about.
Yeah. I don't know whether you'd want to regulate them through our regulation. Yeah, but I agree fully. A single microbe, it's really impressive that it has an effect on an ecosystem when you add it. Yeah.
Okay. Any other comments?
Yeah, this is Anja. So maybe one last question, Anne, before you have to leave.
To cycle to school. Yes.
Yeah. So I was wondering about the pathogenicity that you mentioned. As I understood you, that is related basically to pathogenicity to humans, where we know basically a lot about, not everything, but quite a lot. But what about pathogenicity to non-target organisms, other organisms than humans, be it vertebrates, be it even invertebrates, where we know much less about diseases and pathogenicity?
Yes, it's indeed more tricky. We have a lot of knowledge about pathogenicity to humans, of course, and way less about, well, vertebrates we kind of still know. But for example, to insects or, in some cases we know it's pathogenic, of course, because it's used as an insect pathogen. But the body of knowledge is much smaller on pathogenicity to other non-target organisms. And this is also why having proper test guidelines would give more information to the risk assessment there, because we know less. But it's also very difficult to know, even if you have test guidelines, how to interpret signs of pathogenicity or infectivity in a DEFNIT, for example. How do you do this? So there's a lot of work that needs to be done there. On the other hand, well, of course, for most microorganisms, the non-target organisms are already exposed to all these organisms. And the exposure will be comparable to the natural situations. So it's a bit different where you have the situation of the strawberry box and a child eating the full box. And of course, you don't want to have an effect on any child, versus the environment where you have a bit more leeway in that there should not be any unacceptable effects instead of no negative effects at all for humans. So the risk assessment criteria are a bit different, but it would be very helpful, and that's why I'm very happy that you're here today, to have test guidelines also for microorganisms for ecotox.
So if I understand right, so the non-target organisms are also considered for pathogenicity criteria, but in the absence of proper test guidelines, you just do kind of a qualitative test assessment, it's likely or not, and then basically you approve or not waiting basically for, I don't know.
Yeah, it depends also on the organism. So for some organisms, of course, we know they're pathogenic. So if you take, for example, Metarhizium, which is an entomopathogenic fungus, we know it's pathogenic to insects. We have some strains that were isolated, for example, I don't know where, South America. So the strain was not isolated from the EU, and still they are approved because we know that these strains, the effects that these strains give are similar to the Metarhizium strains that are out here everywhere in Europe. And we know that, for example, bumblebees are used to vector this same strain in greenhouses. So they are used to vector the fungus to the plants in greenhouses, and the bumblebees are fine. So we know they are pathogenic, but we know also that the effects are acceptable.
And you know this from literature studies or from formal OECD, well, there's no formal OECD
No,
guideline studies
yeah,
that
no.
you can know it from.
Yes, no. So there's US EPA and PMRA from Canada, and then they should all be adapted a bit for this specific microorganism to be able to have a test. But it's also from literature, such as, for example, the case of the bumblebees and the vectoring, and that, well, that's of course very helpful to the risk assessment.
Well, that's the problem, I think, with all the literature studies, of course, that needs to be assessed and taken into account, and then you take decisions on maybe pathogenicity and infectivity on all the knowledge on microbiology. But then you can assess only the known ones. You don't know about the unknowns, and you don't even know the unknowns, right? So
I thought it was difficult already, but you've now made it several times more difficult. Yes, that's true.
When we go for the pandemic, for example, we had no idea that such a virus was out there, right?
Yeah, no, but we knew about these types of viruses for sure, and we knew that there might be. So we knew about closely related ones, and we knew that it might happen, but nobody, of course, expected it. So that's different. But there, based on the body of knowledge, we would have said, "Ooh, be careful with this one because this might be nasty." Yeah. So for humans, we know a lot. Yeah.
And we would assess basically on such information the same than for non-target organisms, although we naturally know much less about them.
And testing also, whether we see any effects there. But
Okay. I think we will have to stop here. Thanks again, Anne, for your very nice and clear talk. It was very informative for all of us. Last point, I wish also to invite all of you for the next webinar, which is in 2 months. It's already 28th of July with Stefan Geissen and the role of protists in soil microbial ecology. I know it's vacation during this period, but I hope most of you will be present. And again, I wish you a good weekend. Thanks again. See you very soon. And bye-bye.
Yeah,
thank you.
For the opportunity, and enjoy your weekend too.
And thanks a lot.
Bye-bye.
Bye.
Bye-bye.